Decreasing population size due to habitat loss and fragmentation leads to lower genetic variation. Conservation genomics describes genetic variation within and among populations and identifies the processes influencing this. These genomic data can be applied to minimize the risk of inbreeding depression and increase evolutionary capacity, and therefore improve the conservation of biodiversity in a changing world. Professor Adam Stow from Macquarie University in Sydney, Australia, is an expert in conservation genomics. In his work, he links genomic and environmental data to address research questions in conservation management, ecology, and evolution. He leads numerous conservation projects, such as studying the population dynamics of great nurse sharks and Australian sea lions, and is passionate about education and scientific communication.
Adam, can you briefly explain to us what genomics is and how it can be used for conservation?
Okay, so genomics is an approach to look at the whole genome of an organism. You may not sequence the whole genome, but you're certainly getting information from the whole genome. And you can apply it to a range of different questions. The sorts of questions that I tend to apply it towards is conservation-based questions. I'm particularly interested in using genomic approaches to characterise genetic variation within populations. And the reason I'm interested in that is that low levels of genetic variation limit the adaptive capacity of populations and can also result in inbreeding and inbreeding depression. And one of the major threatening processes in the world today is habitat fragmentation, habitat loss. Populations are getting smaller, and when populations get smaller, they tend to lose their genetic variation. So we can characterise that, identify populations that have lost their genetic variation, and then come up with solutions.
Can you give us an example of how you applied conservation genetics in practice?
Yeah, sure. So there's a number of threatened species that I've been working on. These include the grey nurse shark, which is critically endangered off the east coast of Australia. And so the first thing that we did was work out whether the East Australian population is in fact isolated or whether that population is getting supplemented by migration and consequently gene flow from other parts of the distribution. So once we've ascertained that, we've then looked at the rate at which genetic variation is being lost over time. We've also done similar things with the Australian sea lion and the Great Australian Bite, also endangered, and similar sorts of problems. Yeah.
So, and in conservation management, we could then, for instance, help these populations or also probably bring new species into these populations for diversifying the genetics again, or?
Yeah, so what you can do is you can find appropriate source populations for translocation to bring new genetic variation into populations that have low levels of genetic variation. So that's one of the things that you can do. You can also use the genomic information to look at the features in the environment that facilitate movement or dispersal and gene flow. And once you've identified what those environmental features are, then you can use that information to construct corridors and re-establish connectivity among wild populations. Yeah.
So conservation genetics is a fast-moving field with many new discoveries made every year.
Yeah.
So what do you think? What is the direction conservation genetics is headed to in the future?
Yeah, well, look, I think sequencing whole genomes is becoming cheaper and cheaper. So that with the cost coming down and the efficiency increasing, I can see that we're going to be making use of whole genomes from wild populations, from non-model organisms, a lot more frequently. And I mean, to me, it's just amazing because it wasn't that long ago that that they were struggling to sequence the whole human genome, but now we can apply that technology to non-model organisms in the wild context and get a lot of information for a variety of questions that we simply can't tackle at the moment.
Could you share some advice for early careers researchers with us interested in conservation genetics?
Yeah, sure. Look, I think probably the focus is on big data and the sorts of skills that you need to organise that data and analyse that data. So I guess the general discipline of bioinformatics is an area that will be crucially important to a up-and-coming conservation geneticist. Yeah.
Adam, you have done fieldwork all over the world and in many amazing places. Can you share one very particular memorable moment with us?
Sure. Well, the one that comes to mind is in Amazonia. So I do a fair bit of work in the Amazon basin, and I work on poison frogs there, and also was working with a student who was working on snakes. And we're in a remote part of Amazonia between the city of Manaus and Porto Velho. We're in the rainforest at night, had our head torches on, and it was pouring with rain. And anyway, we turned our head torches off, and we were surrounded by fluorescent fungi. It was just luminescent fungi everywhere. It was magical. I still remember it very vividly. And anyway, turn the head torch back on, and we turn around to walk out. And there is a giant bushmaster snake, curled up like a sombrero hat, this high off, quite high off the ground, 3 foot off the ground. And there it was in the rain. Turn the torch off again, surrounded by luminescent fungi. So it was one of those images that I'll always remember. Yeah.
Wonderful. Thank you so much, Adam.
It was my pleasure. Thank you.